Hello, everyone, and welcome to the Midweek Program at the J.C. Raulston Arboretum. My name is Blake, and I'm the Education Assistant here at the Arboretum, and today we are in the White Garden because there are some lovely plants around us. There's a beautiful Zelkova serrata goshiki here that you should be able to see. In the background, washed out by the sunshine, which is making everything impossible to see, is a big and beautiful Arundo Donax peppermint sticks, which I assure you is fantastic. This is the perfect spot for our location for our program today, because today we are going to be talking about variegation. We will be scratching the surface of variegation. This is an incredibly deep topic. We're not going to get to all of it, but we should be able to wet your whistles about it. We've got our research technician, Evan Villani, here, as well as our director, Mark, as well as a bucket full of beautifully variegated plants. And we're going to explain the science of variegation and show you some interesting variegated plants while we're at it. So it should be a wonderful program. Okay, those of you who are watching this on YouTube, please like this video and subscribe to our channel, and feel free to leave a comment down below. And with the announcements out of the way, I will pass things over to Evan and Greg to teach us all about variegation. Well,
Evan and Mark.
Evan and Mark. What did I say?
Evan and Greg.
Who's Greg?
Wrong director.
That's
Honestly.
all right. I've gotten used to it. I used to work with a mic, and we were Mark. Yeah. At this point, I call my kids after my dog's name, so yeah,
Okay, it's all fair.
good. Yeah, so we're going to be talking about variegation, which is one of my favorite topics. I love variegated plants. In the UK, I'm considered tacky because of my love of variegated plants. They're too proper for variegated plants.
They're purists over there?
They're purists over there. Not everybody, but an awful lot of them. And you know, I'm like, hey, that's fine. There's a whole lot of other reasons I'm tacky to the English too, so it's not a big deal. But, you know, and there's a lot of people who grow variegated plants, house plants, garden plants, and a lot of times they don't even think about it. Certainly don't think about how it works, how it happens. So we're going to try and, you know, we're going to show you some cool plants, but also talk a little bit about how variegation works, and in some cases show our ignorance about what we're talking about when we can't tell you why a certain plant is variegated the way it is. It helps if you know a little bit of science behind that. And
Yep.
like we said, Evan's fresh out of college, so he still knows the science.
It's in there a little bit, you know. So yeah, we've got our educated guesses as to how some things work, but we can tell you the basics, and hopefully we'll break this down in an understandable way so that you in your garden can look at stuff and appreciate it for what it is. So again, variegation is cool. This is a fun topic to talk about. There's 1,000,001 probably different variegated plants, and so we're going to try to break it down to the main categories of it. We'll start real quick by talking about color and leaves. And so we're familiar with the color green. That's coming from the chlorophyll family. And chlorophyll are mostly pigments that capture light and are used in chloroplasts so that plants can make energy out of sunlight. They make carbohydrates using the sunlight. Hopefully we all know this. And then the other families are anthocyanins. And anthocyanins are your reds, purples, pinks, and those hues act as sunscreen to defend the leaf from mutation from sun radiation, and they can also be used to attract pollinators and dissuade herbivory sometimes. There's also carotenoids, and those are your yellow, oranges, and reds, which are used to the side of chlorophyll molecules to transfer light. They help direct light to the main functioning body. It's like, if you look at it really close up, it's this network of all these little pigments, and it's almost like a solar panel, how they direct light to one thing where all the energy is focused and the carbohydrate can be made.
Yeah, and you wouldn't, if you've ever been to the tropics, look at tropical trees, a lot of times their new growth comes out red and often kind of hangs down. I think of a mango tree as a really beautiful one that does that. And then as those leaves mature, they get green, they get stiffer, they open up, and that's, they come out red for that solar radiation, that heat that was mentioned. And something that confuses people about, and we'll talk about this a little bit but like purple leafed plants. They say the purple, it was actually almost in a law in North Carolina that we shouldn't have purple leaf plants because it takes the place of chlorophyll. Chlorophyll is in chloroplasts. These other carotenoids, anthocyanins, are in other parts of the cell, not in the chloroplasts. They mask chlorophyll, but they don't take it away, and the plants can still photosynthesize.
Yeah, the green element is present. It just, when you're looking at it, our human eyes pick up other colors if there's more of that pigment, or the way structurally, like if it's the anthocyanins on top of the chloroplasts, stuff like that makes it appear purple to our eyes.
Yeah.
It's all about how the light gets directed. And it's a little too fancy, a little too rich for my blood to get into all of that. And then I want to talk a little bit about genetics and cell layers while we're here before we dive into it. So a lot of color is influenced by genetics. So we think of variegated stuff oftentimes in terms of mutations, but a lot of plants have naturally occurring variegation because it provides some sort of advantage. And that's like, there's structural elements to that. And then within cell layers, there's 3 main cell layers that we'll be talking about. L1, the epidermis, the outside. L2, the mesophyll, the spongy inside. And then L3 is like vascular tissue. And so that we'll go back over that when we talk about chimeras. Which I think we're ready to get into our first type of variegation, just chimeras, which within chimeras, there's 3 types of variegation, 3 main types.
3 main types.
We'll start, what is a chimera first off?
Great question, sorry. A chimera, that's an old word for multiple things within the same body, right? You think of the chimera, that's the head of the lion, the snake tail, and the goat head also. I don't know why it has 2 heads. They could have given it, off the sides and stuff, and goat hooves. So a chimera is a plant that has multiple genotypes existing in the same body. And so how that's happening is not the entire plant has this mutation, but only a certain cell layer. So first we're going to talk about periclinal chimeras, which is a chimera in which one of these 3 layers has a complete mutation.
So, as Ed was saying, you've got the 3 layers of a leaf, and in general, the L1, the outside layer, is 1 cell thick. The L2, the next layer, is 1 cell thick. And then the L3 can be thicker. Often the L2, that mesophyll, that 1 layer of cells on the inside, can get very large though. The epidermal tends to stay very thin, but the mesophyll, they have, now I'm going back. Palisade cells? Palisade cells.
That are tall.
Tall. So if you've ever been to the Palisades in New York, you know what we're talking about.
It's a palace-like cell. It's very tall, and it's so that light has enough time within the cell to get to the chloroplasts.
Yeah.
But tying it into chimeras, if you've got a non-chimera on those 3 different layers, each layer will have the same genetic material.
If you have the type that, mostly when we want variegation, we want something very stable, and the most stable type is periclinal chimera. You don't have to remember these words, but and that's where one of those layers, as Evan said, is completely genetically different. And with variegation, we're talking either about 1 layer having another
genotype.
Another genotype, but it could be a lot of things. We're talking about color here. Typically, we're thinking of chlorophyll and lack of chlorophyll.
That's your white. White is a lack of pigment in the cell. Yeah. And real quick, while Mark mentioned it, this is useful information when you're shopping. There was a huge Monstera uproar a while back, and what you kind of looked at was how many of these mutations are actually, when you keep the Monstera and you grow it out for a year, how many of those actually keep the mutation? And sometimes they would revert, but a person paid upwards of $100 for that plant. And so this is a stable type, and I'll show you how to identify it. So a periclinal mutation will have a very similar mutation across each leaf. So here we see that the internal section of the leaf is green, and it's very consistent, and the outside is white. And so this is the epidermis, likely has a mutation that causes no chlorophyll production, and then the internal section is still producing chlorophyll. Is that correct?
No.
I'm incorrect. Please.
So on the leaf, even though we say the L1 is the top layer, it actually usually doesn't cover the entire leaf. And so it's actually the L2 layer generally, when you have a white edge, it's generally the L2 layer that you're actually seeing. So let me see these guys. So with the L2, you're looking at the epidermis, but the L2 is going out beyond the edge, and that's the most typical one. Now, if it's the L1, sometimes you can't see it, but sometimes it'll show up as kind of a silvery blue while you have a darker green outside, or it'll show up yellow when you have a darker outside. So because it's the outside layer we call the L1, we think of it as covering the leaf, but it's actually the L2 that you're generally seeing with these. Now, these are two different ones. This is Mygema and this is Rebecca.
Daphne.
Daphne odora, winter Daphne. And if you look at these two, this one, the centers tend to be more muted. And I can't say for sure, but I would actually, going back, I think this may have two periclinal mutations in that the L1 and the L2.
L2.
Whereas this one's just the L1. And what you're seeing is that thin L1 layer kind of masking some of the green that's in there. So it's not quite as dark a green as the Mygema. That's some speculation without getting in with a microscope. I don't know.
Right, and some genetics.
But here is another Daphne odora, and this is only an L1 mutation. So when you get that, what happens is the outside, the L2 is still green, so you get that
green
edge to it. But you can see the L3 in the center there where it's thick enough. Again, I'm mostly sure that's what's going on. It could be another type, but we're going to go with that.
While we're on this plant specifically, do you think that the mutation in the epidermal tissue causes that undulation, that waviness in the leaf?
You know, we're talking about variegation, but it's a different genotype, a different genetics in there, and so it could be causing other issues as well. Plus, if normally the cells would expand at a certain rate, if because of the lack of chlorophyll, they're expanding at a different rate, you can get the green growing more or less. This one first. This is a boxwood, same L2 variegation.
One thing to note here is that instead of white, now you're seeing a yellow, and that's because there's still some carotenoids left in those cells on the outer layer. So there's still in the L2 some carotenoid pigment, giving it a gold look.
And this is something that happens a lot of times, and this has to do with those layers and how they develop. New growth on this is almost pure green, and in some plants it can look pure green, and that's because that L2 layer hasn't really expanded yet. The outside layer is really covering everything, and as it gets a little bit more mature, that L2 layer continues to grow, and you start seeing that yellow edge around there.
And pigments won't be moved into cells until they're fully developed, usually. So this is a nice, I don't know what plant this is.
That is Carostyrax siliphyllus siliphyli is the cultivar.
Yeah, so I would not have guessed that. And so this has a nice silver variegation on the internal side of the leaf. So from what we've learned, I would guess that the L2 layer is functioning normally because the L2, which covers the entire expanse of the leaf, is still producing chlorophyll because you get that green edge of the leaf. However, maybe the epidermis is not producing as much chlorophyll, or could it be the vascular tissue?
It could be. This is one of those iffy ones for me. It looks like it's a L1. This is what I would look at and say L1 because it is very stable and across there, but It could be one of the other types of variegations we're going to talk about.
It's not perfectly stable. This stripe seems to be a lot thinner than this stripe.
Yeah. And, I don't know if that's how the leaf develops in terms of the thickness of the L1, L2, but this could be just a pretty stable mericlinal variegation, which we'll talk about.
Right.
It's kind of same thing with this one. Nice evergreen oak relative.
It's just a nice piece. Anything specific you want to say about this?
No, it's just pretty.
Yeah. And you can say that for a lot of plants.
Edulous starburst. All right. Now we've talked about this being the most stable. So you want to take that?
So
stability.
We've seen this. You might see this. Ligustrum was a pretty popular one that tended to revert, and so a reversion is when basically a mutation mutates out in some way. And so there's a couple of ways that this can happen. When we propagate these plants, when you propagate a chimera, you can't take the seed because, well, for a couple of reasons. Usually gamete tissue arises from only the L2 layer, so you're not going to get all 3 layers of genotypes in the seed. The other reason is that with any sort of adventitious growth, there's a chance that the mutation will not make it back into the plant. So when we propagate these plants, we want to make sure that there's a leaf bud or some meristem tissue that has all of the mutated elements and the normal elements. So we get the regular, we like this, we want to keep this exact same thing. So we take a cutting or we graft it. But in plant growth, sometimes things happen adventitiously, and you can tell as this shoot grew out, it lost its mutated element.
Yeah, and it's really, this is one that we've had for years, Deutzia that's variegated, but right now it's almost entirely green. It has lost almost all the mutation, and in some plants you'll get one shoot that reverts and goes back to green, and it usually outgrow. It's got more chlorophyll, so it'll usually outgrow the rest of it. But this is one where it just seems like the plant is not very stable, and we can go in and keep cutting out the green, but maybe it's just not a good plant.
Yeah.
Got mericlinal and sectorial, and
Couple oddball hollies.
I'm not good at telling what is what on those. This I think is sectorial, but I'm not 100% on that. You want to explain sectorial?
Sure. So sectorial is a type of mutation that a part of each cell layer has the same mutation. So there's 3 cell layers, and a part of each layer has the mutation, but no layer has the whole mutation. No whole layer has the mutation, so you end up with a lot of variety in the foliage. So this leaf looks very nice. It's got a little bit of a white tip, but it's mostly green, and that ranges all the way to these where everything is white, so nothing is producing chlorophyll, and you can't propagate this. This has no chlorophyll. This stem has no way of producing energy. It only exists on the plant because it's basically just a parasite, and the rest of the plant is doing all the work, but it still looks pretty, and it's over there in the white garden, and it has a very nice overall effect. So these are still very nice plants, even though the mutation might not be as stable.
And so what's the difference between this and the mericlinal?
So we'll see. A mericlinal mutation is a part of one cell layer is mutated.
All right.
And we can pull up a mericlinal and show you what that looks like.
So, like I said, I have a hard time telling the difference between sectorial and mericlinal sometimes. So these
probably because we don't have X-ray vision.
Right.
We do our best.
Some of this, when I look at it, it looks like that white is, kind of going through in different spots all the way through, but it is so hard to tell with some of these. So I just brought a variety of things out to show, and that's, just, you can get this speckled, and this is a really very regular speckling of the plant. It's interesting. There are a couple plants that are like this, where this is the first flush. We cut this one back, so this year, so it's just flushing out with new growth, and this is what you get. If we hadn't cut it back, we would have gotten a flush of new growth that looked like this. And then we would have gotten a flush of growth that was almost entirely green with just a little bit of variegation in it. Why? I don't know. We have a loquat that does that as well. There is a camellia that flushes kind of alternately with a marginal variegation, and then the next flush comes out speckled. If you can explain that to me, please do.
So I'll take a second to explain what I think is going on here. So when you look at this leaf, you see just the specklings of green. When you look at the leaf edge, there's nothing particularly special at the edge. So this might be a case where both the L1 and L2 are partially mutated, so that as the leaf expands, random parts of the leaf are still normally producing chlorophyll, but there's large chunks of it that are not, and that look white.
This might be sectorial, because we're looking at all three layers are, in some instances, are variegated.
It could be.
And then you have some lighter and some darker, maybe darker where none of them are mutated, and lighter where two of the three layers.
And if you don't mind, I might grab an aspidistra leaf, or you might grab the aspidistra leaf, and we could also talk about a different reason behind speckling, which is a genetic thing called transposons. I'll take the speckled one, please. It's my favorite type of shopping.
There you go. You got a Cuba and aspidistra with speckling.
So speckling and the way that a leaf expands seem pretty random. And when we think about genetics, we think, well, there's no randomness, it's all planned out. However, almost half of the human genome is made up of transposable elements. And so we can assume that between plants, there's a lot of transposable elements in them too. And those are elements that move around within the genome. So just to get a little bit into that, and then we're gonna get right back out, is that within the gene, it's encoded a protein that allows it to snip itself out of the genome and then something to glue itself back into the genome somewhere else.
So when you're saying genome, the DNA, literally parts of the DNA transpose, they move, transposons, which is different than what we were talking about, where it's the whole cell, the whole, sections of it mutate. This is where parts of the DNA transpose.
And that might be used to explain speckled patterns in leaves. So now we're back into normal territory, and that's where we'll stay for now.
Yeah. And that's why you get things like this. And
yeah,
I'd say, one with the Acuba here, the gold dust plant, you can find this in the wild. Mostly it's green, but you can find speckled ones. But it's different from some of these other mutations, because if you collect seed from an Acuba that is speckled, the female Acuba, if mommy is speckled, the offspring will be speckled. Where, even if you have all green dad and a speckled mom, you would expect, based on typical genetics, that you would have 25% speckled, 25% green, and 50% somewhere else in the middle. And you don't. You get 100% speckled. Kind of interesting. We don't have to go through all of these, but here's another one that perhaps is sectorial.
So that's,
where you can see it's,
again, you're getting those leaves where there's no chlorophyll production, all the way to the range of some oddly shaped leaves as well. There's actually 3 colors in this. You see full chlorophyll production, the dark green, probably just one layer of chlorophyll production, the light green, and no chlorophyll production, that kind of yellowish white.
Yeah. And to go back to the very beginning, it flushes out kind of pink usually, especially on the white ones.
Oh yeah, you can see a little bit here of those anthocyanins, and so those guys are doing the job of protecting the leaf while it expands from sunlight radiation.
And the reason you see that is there in the green leaves. It's just masked. When the green leaves come out, they're all green. You can't see those, but those anthocyanins are there, carotenoids, whichever they are, and they don't show up to protect the white growth. They are there, and it's just, you can see them because there's no chlorophyll to mask it.
Right, and as soon as the other stuff moves in, you can't see it at all.
In contrast to this, and more like this oak, is this osmanthus, where it is very, very regular, the speckling on there. There are almost never any reversions on this Goshiki osmanthus. And I want to contrast that with this Stachyurus, which is can be all over the place. You got some of those solid white leaves, no chlorophyll, that are like parasitic, basically, and the speckling. And kind of the cry of the plant breeder is they want to stabilize the chimera. You've got a mericlinal chimera, you want to stabilize the chimera, turn it into a periclinal, or at least something that is not gonna keep going. So you would look on this plant for leaves, branches really, that are showing more of that edge variegation, and you would propagate that, and then you propagate the best ones from that, and propagate the best ones from that, until the mutation has settled into that L2 layer that gives you that external variegation, or it has settled into the center and you get a center variegation. Sometimes that happens, sometimes it doesn't happen, but that's the thing. They try and stabilize the chimera. Yeah. I have certainly asked Sophia to propagate the ones that are closer to being marginally variegated, because I would like us to get one, but we haven't gotten it yet.
And that could be a little hard to understand, so I'm going to explain it one more time in a slightly different way. So let's say hypothetically a layer is made up of just 10 cells. And so in this sectorial chimera, maybe it's like 4 or 5 cells in each layer are mutated, so they are not producing chlorophyll. But there's a little bit of randomness based on cell expansion rates and just development. So there's some random elements there. And so one branch might have 7 out of 10 in the epidermis layer that are mutated, and so that one looks a little different, and then you propagate that, and when you propagate it, you're starting from a new base where 7 is like the norm. And so if you're it's like a group of 10 friends, right, and 7 of them are doing this, you're like, well, I'm more likely to stay around this area. I don't know if that makes any sense, maybe not. But then from 7 you might get 9, and eventually the goal is to get 10, because once you have 10 out of 10, that's the one genotype in that layer. And so there's a very low chance that anything else is going to happen, basically.
There you go. Now we got some other types of variegation go on. Let's you want to start with?
Yeah, let's clear some space here. Asarum, this is beautiful. So I hope this picks up on camera. You have a light green kind of halo in the center of this leaf. And then dark green edges and centers. And so this is not a chimera. This is completely heritable. This is completely normal. You'll see in Asarum a wide variety of this type of variegation if you just go out and look at a population. So this means it must be conferring some type of benefit. It wants to look this way genetically for some reason that I'm not privy to, because I'm not a bug or a bacteria or something. And so this is purely genetic. And then these are also beautiful. Begonias, one of my favorite plants. I love the hardy begonias we have here in the garden. Beautiful colors on these. What's happening here is that if you look through, you don't see that white, so it's not totally translucent, but you're seeing this white on top. Now what's causing this is actually a structural effect called blistering. And that's when the top cell layer, so if there's a flat cell layer underneath, the top cell layer does like this and traps air underneath it. And actually what you're looking at is a reflection of trapped air within the leaf. And so again, this is genetic, so it must be conferring some type of benefit to the plant for it to have this silvery white look.
Or at some point convert a benefit that may or may not still
it's true, yeah.
hurt it.
Could be an evolutionary, backlog, or what's the right word?
Vestige. A vestigial trait.
That's right. There you
Great
go.
word. Blake's great with words. That's one thing I've learned while working here.
All right. Now, maybe you understand this type of variegation better than me. And already during this, I've already changed my mind on what I think this is. I was thinking this may be a blister, but it's not. This you sometimes get is this variegation based on around the veins. Now, you can get this kind of thing from nutrient deficiencies. Iron deficiencies often show up as yellowing between the veins, not yellowing on the veins. But this is a natural, not a natural, but this is a mutation that is incredibly stable. And here you have kind of silvery blue with the veins staying green.
So my guess for this forsythia is that since you're basically only seeing this on the new growth, right? Is that there's a different structure above the veins. And so it could be one, that the structure above the veins is different than the rest of the leaf, as in maybe the other parts are thicker and can fit more chlorophyll, or two, the cells that are between the veins expand and mature first, and so chlorophyll is being sent there, and the cells around the veins are still expanding and maturing, so there is no chlorophyll there. And so that gives it this following the venation yellow pattern.
Yeah, could be.
You know, it's just a guess. The fun part, right?
Yeah.
I don't know, because this is a mature leaf and the entire plant looks like this. This is on the wall of McSwain? Is this one?
No, this is in the Japanese garden. This is schizophragma, or now hydrangioides, technically. Climbing hydrangea, pains me not to say a hydrangea relative, called Moonlight. And this is how it grows, with this bit of shimmery, and it could be that it's the outer epidermal layer does go all the way to the edge and is very thin, but doesn't have chlorophyll, and so it's muting the green color, but the cells below that are thicker along the veins and they show through, but I don't know. But hey, wanted to show. I'm not afraid to say I don't know. So monocots, these are the grass family, but the monocots include things like elephant ears, aroids, ericemas, things like that. Here we have a rhodia, a corn, which is actually where the first example of transposons were discovered in corn, the corn kernels. Here is a gingiber. Here is a carex, so more of a grass, but it's also, think elephant ears, and some other plants. Agaves are all monocots.
And so to contrast, this is a dicot leaf, variegation, and to my understanding, the pattern of variegation in monocots, that striping, how it goes often laterally, or you might even see it horizontally in some miscanthus.
Yeah, that's very rare.
Very rare.
That you get that, because the veining goes long ways, and so you almost always get an edge
going down the leaf,
or
along the
striping,
leaf.
or this kind of thing. But there are ones that do go across, and plants are wild.
It's just a different leaf structure between the dicot, which can have it all the way around a leaf, versus the monocot, which often stripes down the leaf.
Yeah.
Around versus down.
Ferns, I don't know.
Right, because there's an arthurium with like 3-4 different colors.
Yeah, so there's a kind of a weedy, an arthurium we have in our laugh house that came in as a weed on another plant, that's called snow cream, and it's got multiple colors, and it's kind of splotched through, more like a sectorial or a mariclinal. This is Coniogramma emansi. This is one called golden zebra, but I have seen this on Mount Ime, where it's from, and it looks just like this, with these light areas on the leaf. I don't know quite the reason for that. And tropical ferns, there's a lot of tropical ferns that do that.
I don't know if this will show up on camera, but even the underside of the leaf is beautiful. Nice.
Quick word about, mentioned purple leaf plants, and if you go to England, you go up to the Northeast, and you look at a purple-leafed redbud. And this is one we introduced, Merlot, with Denny Werner. It's not real pretty right now. And the reason for that is the purple is in the L1 layer on this. And in England and the Northeast, they don't have the intense heat in summer we have. And the L2 layer and the L3 layer don't get as thick. And so the green, the chlorophyll that's under the L1 layer doesn't show through as much. But almost every purple leaf plant, that woody plant that we grow, the purple's in that top layer. And so you get muddy color by the time it gets through the season. This Merlot was bred with the Texensis type, which has a thicker leaf to start with more so it has more of that L3 layer in there. So it really gets muddy, but it stands up to sun much better than like the old forest pansy. But that got muddy. This gets muddy even sooner, unfortunately. Comes out beautiful. Comes out dark purple because it's really, those other cells haven't expanded. So, but that's, you think those layers. This is one that I'm really liking. This is a styrax, a snowbell called Evening Light. Now this is one that comes out greener than like the Merlot. It really comes out fairly muddy green and actually gets darker as we go into the summer. So it flowers after the leaves come out. So when it flowers with white flowers, it is almost black purple. And that's because it's the L2 layer that has the pigment in it. So when it comes out, there's more of that L1 layer showing. So that was L1 was purple in the red bud, so it comes out very purple. This comes out more muddy green. And then as the L2 layer expands in early summer, it gets very purple, dark purple. And then that L3 layer builds layers. Instead of just expanding, it gets more and more. So as you get into late summer, it loses more of that purple. But if this were on the L1 layer, it would be almost fully green by now.
Yeah, it's always fun to watch a plant through the whole season.
Yeah.
And see that kind of change. It's a beautiful plant from any distance.
So if you want something that stays purple all season, what you need is L1, L2, and L3 to be purple. So just ask your retail nursery, what layers of the leaf are actually purple and which are not.
Yeah, and it might be good before we wrap things up here to talk about the most applicable elements of this, which are if you're propagating a variegated plant, it's good to know, maybe guess what kind of variegation you have. If you think it's heritable, sure, go for it. Prop it from seed. But if you think that it's a chimeric mutation, you're going to want to keep a leaf bud. So you take a cutting or you graft it. And then know what you're buying as well. If you have a specific plan, maybe take a second before you get caught up in a houseplant craze to evaluate if this tiny little plant, when it actually grows 2 more leaves, is still going to keep that trait. Or with landscape plants, it's usually, they're pretty established.
But even those, you want to cut out reversions, things that go back to green, things that look different. And by the same token, if you have a green plant and it throws a variegated shoot, a mutation, hey, give us a call. Sometimes those things are really cool. We'll propagate it for you and give it back to you. So I want to end, we talked about these transposons and these acuba that do this, like that have these regular spots that occur naturally. Well, doesn't mean they always have to do that. We have here some sectorial variegation in addition to those transposons. We have this, what do we call this?
What species?
There's another acuba. It's just
Japonica?
Yeah.
Really?
It's just the whatever the mutation has made the leaves smaller, narrower, and they're all speckledy and whatever. So
interesting.
Mariclinal or something like that. Here you have one that's L2, got that really common thing, and this is coming off a mutation off of a solid green leaf plant. It's not a speckled one. This is a little harder to see. This has central variegation. Now this is actually in the L3 layer. And if you grow it in full sun, this gets really bright, much golder. It doesn't get this bright because this is gold all the way through, but that gold shows up through the green much better when you get more sun because those layers really build. And then you have this, which is got both the transposons, but also this sectorial variegation that goes, it's all 3 layers through parts of the leaf. And I feel bad about taking this off because this is a painfully slow growing plant because
it's a lot of white.
Look how much doesn't have chlorophyll at all in it. And it reverts a lot. It does not stay this way very well. And it's called Mangetsu improved because it reverts less than the old form that was out there. But you can see it's got this white stem as well. It doesn't even have a green stem to it. So it's it's really it's a cool landscape plant. This thing would be gone in a heartbeat if it were out in the wild. It just couldn't compete. So a mutation like this would die off. And that's a lot of what horticulture is finding genetic mutations and keeping them going when, they would probably die off in the wild.
Yeah.
I don't know why in the tropics we have so many more variegated plants naturally growing beyond just red. What
advantage do they or did they confer?
Yeah.
Is interesting. Probably someone is probably studying that.
Probably.
There's a lot of people out there.
It might just be a function of the greater genetic diversity of tropical areas generally. Like the closer you are to the equator, just more diversity is there. So with more diversity, more chances for mutation.
Yeah.
Before we get too carried away, we'll take some questions.
Oh yeah, absolutely. We got a bunch of questions, so let's get cooking on them.
All right. Ooh, that's a lot of questions.
Okay, yeah, that's what I'm saying. Okay, so Greg wants to know, is the variegated Daphne odora more tolerant of wet conditions?
No.
No. Okay.
But
Daphne's still Daphne.
I feel like it is I feel like these are better growers for me than the all green leaf one. And there's a reason, and it's not just because people like variegated plants that you can, if you find it, you usually find Daphne odora aurea variegata, which has a thinner, it's creamy yellow margin, but then it gets white as the season progresses. But you see that more often than the green leaf one, which if anybody's growing the green leaf one with white buds and flowers instead of the pink buds, that's one I want. I want the green leaf with the white flowers, but you hardly ever see that.
Awesome. So Carol says that she has a variegated redbud tree that shows a different level of variegation from year to year. Some years it has a lot of variegated leaves, other years not very many variegated leaves, and it goes back and forth. Do you have any idea what would cause this change from year to year?
No. Now, I will say we had two variegated redbuds across from each other, not ones that we had introduced, but one was floating clouds, one was silver clouds, and I can't remember which was which, but one of them, the variegation was really bright when the leaves came out in the spring, so it was on an outer layer, and it was splotched. It was mericlinal, not an edge, but it would come out really bright, and the other one would come out almost pure green. And as the season progressed, the one that came out green would get more and more variegated, while the other one would get more and more green. So it's kind of just interesting to see two plants that at one point in the summer looked almost identical, but you could tell them apart spring and fall.
Yeah, based on the differences year to year, it could, again, there's a lot of random elements in genetics. It could be how the leaves are expanding. It could be like the week that they start like forming a bud.
It could be as simple as if it's a hot, dry summer, then maybe the cells don't expand as much. That maybe it's a lot of the variegations in the L2 layer, and those palisade cells don't expand as much. They're tighter, and so you don't have as much of that area of the leaf with the thickness of the leaf with no chlorophyll, or it's really a wet summer. They really expand more. You get more growth. You get more green that way. It's hard to say.
Sure. It's a complicated topic.
It's a complicated topic.
Yeah, hopefully we have explained it well.
Yeah.
And I appreciate you starting this all off with scratching the surface of it.
Yeah.
Yes.
It is complex, and there are other variegations. There are viral things. We only talked about foliage. There are variegated flowers.
We only had an hour.
Yeah.
We still got questions, y'all.
All right.
Kathy wants to know what the name of that speckly oak was.
I got you get that?
There's two oaks that were speckly.
This one here.
This one. This is Quercus robur, so the English oak, First Schwarzenberg, F-U-R-S-T, Schwarzenberg. It is one of the most difficult trees to graft. A great grafter. We got this tree, I bought it at an auction that we were putting on. The grafter who gave it to me told me how difficult it was and said it better go for a lot. It did. I paid a lot. And then we brought it here.
That's funny.
It's I gave it to the Arboretum. Yeah. So you don't see it often. We actually sold this past spring or last fall.
Okay.
I can't remember. We sold this in one of our plant sales, and almost nobody bought it. Which I was like, well.
Well, there's no accounting for taste. I suppose.
This one's a pretty. Is it short?
It's short right now because we cut it back to the ground.
Because of the reversion, or why?
This is something that happens a lot of times with variegated trees. Is when they're young, you see a lot of the variegation, both because it seems to be a lot brighter and because there's a lot more new growth on a young plant. As the tree ages, one, it gets big and gets above your head, so it's harder to see. But also, instead of putting out a foot of new growth, it's putting out a couple inches of new growth, and you just don't get the effect. So while it'd be awesome to have a big tall tree that looks like this, the big tall tree really looks mostly green.
Mostly green.
Okay. Well, that's fun. Sounds like a good tree to coppice in the springtime.
Yeah, and I do have, here we often leave plants pretty natural. I do at home with some, like I have variegated Elysiums. I have one that flushes out purple and then it's lightly variegated. It's Murasaki no Sato, which we often distribute. That new foliage comes out just dark purple. Well, I let it flush out and then I trim it hard and then it flushes again burgundy, and sometimes I'll trim it again and it flushes the third time. So right now it's flushing at my house right now with that purple, whereas normally it would just be green with the vaguest of variegations in it.
That's awesome. Okie dokie. Ranjani wants to know what is a good variegated dogwood for North Carolina. She says she's tried Summer Fun, Wolf Eyes, and variegated Stellar Pink, but none of them survived.
So those are all Coosa dogwoods. Wolf Eyes is a kind of one of the old classic ones. It's a really good one. Summer Fun is a pretty crap plant. It's beautiful, but I can't keep that thing alive either. Gosh, there's one that's very similar to Wolf Eyes that I'm blanking on.
Would it be better to look at another species? Like if you're looking for a variegated Cornus goat for Florida or fishing?
The variegated Cornus Floridas can be tough. They tend to look a little more ratty at the end of the season than a regular dogwood, but there are some. Actually, some of the weeping ones, Kristen Lipka's variegated weeper is pretty nice.
What about Cornus alternifolia W. Stackman?
So I love that plant. And I just actually the other day went by my old house where it's still growing beautifully. Cornus alternifolia has some issues sometimes with canker.
Okay.
Greg doesn't ever want to plant one here because he's like, it's going to die of canker. I actually think the variegated ones are better of that. The other one that's really good and really brightly variegated is the cornelian cherry Cornus Moss variegata. It is just showy as can be. But that one, you won't get the big white flowers, but you'll get gold flowers early in the season. I am blanking on the name of one of the Cornus Coosa that does really well with variegated foliage, and it is on the tip of my tongue, and depending on how many other questions we have, it may percolate up.
Well, let's see if we can stoke Mark's memory while we answer some other questions. So Carolyn wants to know if we can propagate plants by leaf cuttings, and if so, how?
Not anything woody.
There was a begonia you brought up.
Yes, there was.
That would do with leaf cutting quite nicely.
Begonias will do with leaf cuttings. Eucomis will do with leaf cuttings. Amorphophallus will grow from leaf cuttings.
Huh.
Many of them, not all of them. So here's the weirdest. It's not exactly a leaf cutting, but there is a way to root pine fascicles. So the little bundle of pine needles.
Sure.
You can root that.
I read about that.
I've never done it, but I've heard you can do it.
Yeah, same. As soon as I read that, I was like, what? I'd love to try it. I haven't, but yeah.
Me either.
And so what you're going to do for leaf cutting, just real quick, is you're usually you can leave a little bit of petiole and you just cut that there, put a little rooting hormone there. If you want multiple but smaller clumps, you can cut some of the veins because those also have a lot of that tissue that's going to develop into new types of cells. You can cut the veins if you want that. Otherwise, I would just go for one cut there, rooting hormone, put that in the soil, or not soil, but your rooting media.
Yeah.
And then keep that misted. Because leaf cuttings lose water like no other cutting. Yeah.
Yeah, that's how I like to do them. I like to do them flat, and I do often. I'll either cut veins in here because they'll root there, or else I'll just cut the leaf into a few pieces and put down, little triangles of begonia leaf on there. And you want it to have good contact, so little staples or something will help keep it down, or
pebbles.
You just put weight
paperweight.
Honestly, was Summer Gold the cornice?
Summer Gold was it.
Summer Gold was it. Thank you, Kathy.
That's a very good one. Yeah.
Cornice quos, gold.
Okay, so Greg also has another question about acanthus mollis. Acanthus mollis versus acanthus whitewater. So with whitewater being variegated, he's wondering, does the variegated one need the same amount of sun exposure, or can it take more, or does it need less?
It can take less. You're growing whitewater, the variegated one, more for foliage than flower. So if it doesn't flower as well in more shade, but if you're growing it for that foliage, if you go into more sun, you're going to need to make sure it gets moisture. Something I did not mention that I want to mention with variegated is some plants the variegation does not show up well early on. So often variegated acanthus, it takes it a year or two to show. There's a fatsia called spiderweb, which the veins are white, which is the weirdest thing, because if you grow seed from it, they are all the same. They're all spiderweb. But that takes a couple of years to go. And often that's because the variegation gets masked by chlorophyll, because in the nursery, if it's being produced, it's being pushed with water and fertilizer to get the quickest growth. And so you get this green plant. You plant it in your landscape, you don't over fertilize, you leave it alone, and 2 years later, it's beautiful and showy.
Yeah.
If you can live somewhere where you can grow horseradish, the variegated horseradish, it takes it 2 or 3 years before it really gets variegated in the garden, no matter what. And that's one that you have a big, beautiful clump. You dig out a piece of the root and you plant it somewhere else. It'll go back to being green for a couple of years.
Cool. That's amazing. It's so weird. So Kathy says she has a blondie euonymus that has 4 stems with totally yellow leaves. So it's a sectoral variegation. So she's saying she wants to know, so she wouldn't be able to successfully propagate that characteristic specifically.
No, you can't propagate just the yellow leaves because there's no chlorophyll in there and it cannot sustain itself.
Sure.
Blondie is gold enough that there might be some. There are, you think of a plant like Elysium, Florida Sunshine. There is enough chlorophyll in this to survive.
Yeah.
And I don't know with Blondie, those euonymus tend to be pretty darn unstable. I'm sure somebody has tried to grow them. We have right now, trying to root in the greenhouse, several all gold sports of a Cuba. I have tried a million times. You would think that there would be an all gold one. There is not. And we keep trying. Different from sports from different ones, because they can be wildly variable.
And I think that should be the takeaway, is just try it, because there's a chance that it could work, even though the science would suggest there's no chlorophyll there.
There's exceptions to every rule.
Yeah, exactly.
I
just needs a little bit.
I didn't get to this, but there is a thing called virescence and albescence. These are 3 virescent plants. Virescent means they become green.
Okay.
So this is an Elysium. It flushes out white. Well, pink, because often these white things have that, there's carotenoids in there, but then it will go completely green. The whole thing will be green. Through the winter, it'll be all green, and then when new growth comes out, it's pure white. This is a podocarpus called kinme. It comes out more yellowy than white, but then it goes completely green. There's another one where if you shear it before the new growth comes, you get more new growth and it's very showy, whereas if you just let it grow, you get almost none. This is a hybrid hydrangea between our big leaf and Japanese hydrangeas called Summer Frost. Terrible for blooming, doesn't bloom for squat, but the new growth comes out white. There's an Akuba like that I would have taken a cutting of, but it's solid green. But Natsu Nakoma comes out, and from across the way, it looks like it's this big white flowers on this
shrub because the new growth is so white. No kidding. Really extends that season of interest too.
Yeah.
That is awesome. Okay, and I will say we are over time, but all of us here are doing what we love. And if you all are still loving this, then I encourage you to stay with us. But otherwise, go about your business. You don't have to be here. Okay, so Carol wants to know if you could explain how a sport occurs.
So a sport, which is just when a plant throws out something odd. It could be variegation, like we're talking about. It could be, a different kind of growth habit. There's a lot of things it could be. It is a genetic mutation. Something has mutated in the plant. When you've got something that's already doing something weird genetically, you're more likely to get a sport. This plant probably, if I looked around on it, I would have had a whole branch that was solid gold that I would ask Sophia to propagate. So that's a natural thing. It can happen. You can get a mutation. Can sometimes weirdly be caused by insect damage. You get the witch's broom effect that can come from insect damage. You can make it happen. You can irradiate plants, seed or plants, and often get a mutation. There's also this weird thing where a pre-emergent herbicide, surflan, I don't remember the chemical name for it, surflan, you can actually soak living growing plants in surflan and get them to mutate.
No kidding.
Now, not always, and it doesn't always come out with something good, but some interesting plants have come out that way. And, you can, traditional breeding is not mutation work. That's something where you can bring in new characteristics and you can change the genetics, but it's not really a mutation. Tissue culture, often when you do tissue culture, you often get unusual things.
Lots of damage. So I'll say simply that mutations are happening all the time. You get sports, as in like an entire shoot that has the same variegation or pattern, because when it's only a little stem bud and there's not as many cells, a small mutation can have a bigger impact. So if it's affecting enough cells, then you'll get that eventual sport that shows that. So it's a mutation that happens at the right time as well. And the final thing is that mutations happen all the time. Most of them we don't see. The vast majority of mutations are invisible to the naked eye, and that's just something that plants and everything does and is happening, and we just don't know about it.
Or it's not viable. Something mutates and it dies before anything happens. You've got the tissue just aborts, and then the plant goes, or organism, person, whoever, goes on just fine because it doesn't really affect anything, but it's also not viable. So
Cool. Okay, Sandy wants to know, do these variegations / mutations, etc., apply to the amazing diversity in hostas, or is there something else in play there?
Yes. So hostas, those are variegation, and all that variegation are from mutations. Interesting, if you go look at a hosta breeder, they are not taking the best hosta and crossing it with the best hosta. That's not what happens. They're taking really good hostas and they are crossing them with these ugly things because to get that variegation to transfer, and they're taking the beautiful ones and crossing them onto the ugly ones. And you'll have these more sectorial mericlinal variegation where it's not a margin at all. It's kind of plant actually looks really ugly. But if you look at them when they're going into flower and going into seed, that seed stalk will be variegated up the seed stalk, and the seed pods will be variegated. If it's not variegated, if the pod's all green, they don't even harvest it and sow it. Pod's variegated. You don't get that on those margined ones because it's different tissue that's giving rise to that. The big thing in Japan now with hosta are these thick textured leaves. They're almost like corrugated. I haven't really seen much of anything like it in the US, but there was a lot of breeding work, and that's what people wanted to show us when we were visiting there last year, was in the nurseries.
Yeah. So basically what you're saying is they're crossing to an ugly thing so that they can see which seeds will have variegation, and breeders do that because it's a numbers game, and you don't want to sow out 1,000 seeds if only 5 of them are going to be variegated, because that's a lot of work. So you're getting
the nice ones,
a higher concentration of
the
variegated
nice ones.
seeds.
It's kind of like the Acuba. All female spotted Acuba will give rise to spotted Acuba. If you cross onto that really pretty plant, that mutation is not going up that flower stalk at all. You won't get variegated plants off of it. And if you sow seed in general, you are, say this is any plant, Acuba or otherwise, you have this kind of splashed variegation, you are more likely to get something variegated out of it than you are with this. We think of these stable variegations as, as really great plants, but they're much worse for trying to breed for that, because it is this genetic mutation, and it's in a portion of the leaf that does not become
a gamete, a sexual reproducing part.
Right.
Right.
Cool.
It cannot confirm.
This is so much fun. There's just so much to learn. Okie dokie. We'll do, let's say, 2.5 more questions.
Hey,
Sure. Two of these questions were vaguely similar, but also incredibly dissimilar at the same time. So Dahlia says that she has many ferns, including Japanese holly ferns. Most of the holly ferns are beautiful, deep green, yet a small batch of her holly ferns have some yellow variegation, which starts in late spring and early summer. She wants to know, was this an accidental variegation during propagation at the original nursery? Which is similar to a question that Greg had about his spiderweb fatsia, where he wants to know if he's seeing a bunch of variation in the patterns of the variegation on it, and he wants to know if that's the result of the propagating by cuttings or if the unstable mechanisms are the culprit.
Second seems to be unstable mechanisms.
Okay.
Yeah, although that's something I haven't really seen with spiderweb. I'd be interested in him sending pictures of it. The holly fern, so if the ones that are turning yellow, if they are really growing in the same conditions as your all green ones.
Yeah.
So it's not nutrient deficiency.
So it's not a nutrient thing, a moisture thing, just different soil type thing. It'd be interesting. I don't know if
I've ever
it's pretty
seen it.
yellow. Now, I have at home some really ugly variegated holly ferns that I've gotten in Japan.
Collector's item.
They're very much collector's items. They're very hard to keep alive and very ugly. Something like what you're talking about could very well be some sort of a mutation that's occurred, and people just look at it and say, "Oh, I'll throw some fertilizer on it. Hopefully, it'll be good next year," and they aren't really paying attention to it. So, I'd watch it over time, especially if it's growing in the same spots, and if it's interesting and nice, you could grow it. So for holly ferns, most growers are either, if they're not buying in plugs that came out of tissue culture or from a fern specialist, they're dividing their holly ferns. Otherwise, it's you're doing them from spores, and fern spores are so weird because you can get hybrid ferns, but generally ferns are the spore are coming off of one plant, and the only time you get cross germination is not germination after they form their first structure. If they're growing close together and you throw water on them and shake them around, that's when you can get some hybrids.
Come to our fern lecture.
Actually, we do a fern propagation workshop in October where we go through the preposterous life cycle of ferns. It is utterly dissimilar to every other plant, and if Mark sounds like he's struggling to make sense of it, we all struggle to make sense of ferns.
And it just wasn't preposterous 100 million years ago. It was the norm. Ferns were,
Yeah.
No kidding.
But if the color on those holly ferns doesn't make them look sick, and it's something interesting. Again, I'd be interested in seeing pictures of it. It might be something worthwhile. Probably neither of these are worth patenting and making money.
Sure.
But we love propagating cool plants and getting them out to other people.
Make 10 or 20 and send them elsewhere.
Honestly.
Yeah, we'll let you name it, if it's
Definitely.
something you from your yard.
for sure. Sometimes these mutations do happen in people's gardens, and like every single one of these cultivars got to start somewhere. So this could be.
And I will say with the Fatsia spiderweb, if the grower doesn't know a whole lot about them, they're doing them by cuttings. Fatsia roots very easily, but there's very few cuttings on it. It's a chunky plant. It's hard to make a lot of plants.
Yeah.
So if you get it at the farmer's market in a 3-gallon, it was probably a cutting. But like I said, if you grow seed out from it, it's basically 100% no variation. But there could be some, and that could be what's going on with that.
Yeah, sure. Okay, this will be the final question. Carolyn says that she has an Andre the Giant Ardesia, which has white edges, but it keeps throwing off green sports. She wants to know if she should cut the sports back to the stem of the variegated parts.
Well, so Andre the Giant is green.
Okay. Well now. Interesting.
Now, we have found sports on Andre the Giant. We actually named one that was a stable for Ardesia japonica margin that we luchador, El Luchador, in honor of Andre's wrestling, but it's a little bit over the top and gaudy like luchador. If you got it from us, it might have been, we distributed some before we named it. We might have called it Andre the Giant Sport. And so if you want to keep it variegated, get rid of those green sports. I grow a lot of Ardesia japonica. I love them. Some are very stable. Many are not. And I regularly going in there and ripping out any of the all green shoots from those.
Yeah, and that's generally the approach for managing reversions for all.
Yeah, and with woody stuff, if you don't do it regularly, there are trees we've taken out because we just lost too much of the variegation.
Yeah, it would require taking too much out of it. The plant would look ugly.
Yeah.
But pruning trees is so much fun.
Oh, it is. Yeah. Only if you end up with a good end product.
For sure, yeah.
One last thing, since we're talking about pruning and Japanese maples, variegated Japanese maples are extremely prone to losing their variegation if they are not pruned regularly every year. Some can be cut back kind of hard and you can regain that variegation. Some you can't. The same thing's happening with the other speckled oak we have. We should have pruned it more. We're probably going to look at really cutting it hard, seeing what happens, seeing if it comes back up with that variegation. And that's, like I said, we try to grow plants naturally so we can talk about how big they get. But there are some plants that, to be a garden plant, have to be managed a bit more.
Sure.
Yeah, otherwise it just loses the trait that makes it anything interesting.
I've started growing more like larger maples and larger trees that are variegated, which I was always like, I don't have the space for them. But I visited a garden in England where there's somebody who's growing so many of them, and they grow them as little, lollipop-headed things or prune them. And I used to always be not much of a pruning person at home, but I'm starting to do more and more. And I'm getting some really good effects from doing that. Some interesting garden effects.
And you also started watering your plants too. What's going on with you, Mark?
I don't leave my house anymore.
Oh, okay.
Ever since COVID, I try not to leave my house except for to go to work. So I have more time in the garden.
Yeah, for sure. That's great. And I just want to say, Evan pointed this out to all of us on our Plant Lovers tour this past Tuesday, but this Zelkova serrata goshiki, being as huge as it is, the effect that you get looking up through the leaves when the sun is over them, it is so beautiful to see every single leaf have this halo around it. It's just like a really great effect. And, it is a big honking tree, but it's certainly worth the space.
This is one of the better shade trees, variegated shade trees.
Yeah.
We had some big almost blizzard that's got a white edge. When it's young, it's real showy.
Yeah.
When it gets big, it's not so much.
No kidding. And the almost has kind of like a patchy canopy, whereas this one's real full. Like it just really fills the sky up with that beautiful characteristic.
It's a great camera. Did you see it?
No kidding. So yeah, thank you for pointing that out to all of us on the Plant Lovers.
No problem.
It's a great tree. And thank you, both of you, for being here and going. Well, I wanted to say a deep dive, but again, we were just scratching the surface on variegation. It was obviously an incredibly complicated topic.
Thank you guys for joining us and following along, and now you can tell all your friends.
Absolutely.
right.
Yeah.
And call us if you get a good sport. Send a good
for sure.
picture.
Share it. Absolutely. Share the love. So thank you everybody for joining us on the midweek program today. While I say that, while I look up what we're doing next week, where are we? What month is this? Okay. It's August. It's August. And next week, we are going to have Amanda Bratcher on the program, who's an extension agent in North Carolina, who is real big on pollinators. And she's going to be coming out talking to us about the plant pollinator census, which is going on next week. So it should be all kinds of fun. We hope we will.
She just won the American Horticulture Society's Great American Gardener, one of their Great American Gardener Awards, the worst named awards anywhere, the GAG Awards, but it's a national honor. So,
So that'll be awesome. We will see you all next time. Y'all take care.